All right, we're going to continue our discussion of the origin of biopotentials. Today we're going to talk a little bit more about some specific biopotentials. So in this course, there are three different types of biopotentials that we will look at, two in a little more detail. The first is the biopotential that originates from a muscle, a motor neuron package, often referred to as a motor unit. Many of these combine to give you a signature whenever there's muscle activity present. And so we refer to this as electromyography, this measurement, and we're actually measuring the activity of the nerves when contracting a muscle. The signal from the heart we refer to as electrocardiography. It arises because there are electric potentials that occur that make the heart muscles contract and beat. So in many ways, it is also a muscle type signal, but it's different from the typical muscle motor neuron combination because that would be sort of more your skeletal muscles that control movement, whereas the heart controls obviously the pumping of blood through the body. Movement, but a different type of movement. Also, another difference is that one of these is voluntary. Typically, when you contract a muscle, that's usually a voluntary activity. The heartbeat is referred to as involuntary. The body just does it without your input or manipulation, hopefully. And finally, biopotentials that originate in the brain due to the activity of neurons communicating with one another and communicating with the rest of the body. This can be measured with electroencephalography or EEG or magnetoencephalography, which is MEG. So these are the three main biopotentials that we'll discuss. We'll go a little more into detail in muscle and motor and brain because those are the two areas that I'm more familiar with. The heart, there's a great deal of research and some references available on this. We'll talk a little bit about it, but I'm not an expert in the heart or in the ECG, so we won't discuss it in quite the detail that we'll discuss the other two. I want to refer you to some references, and this is extremely important because many of these references are have to do with some of the slides that I'm using, and some of them are taken directly from these references, so I highly encourage you to go to the actual source. In addition to this, I'll post a couple of papers as well that give good overviews of the different types of measurements. So today in this lecture we're going to focus on electromyography, and we'll look a little bit at the biology as well as the engineering associated with this. So there are lots of things involved in neuromuscular function. It's called neuromuscular because there's communication that goes on between nerve cells and muscle cells. And this is just some vocabulary, different things that you'll see referred to. We will talk about some of these. Some of the other things that get more into the microbiology, we will not discuss. So looking at some basic vocabulary, obviously we know, most of us from a basic biology class, that a nucleus is the control center of the cell. Dendrites basically carry a signal to the cell body. The axons will carry an impulse away to the next neuron, muscle, or gland. Typically in the neuromuscular system, these are covered in myelin, and the myelin acts as kind of like an insulation. Changes the impedance of the transmission path, as well as provides some protection for the neuron. The cell body is referred to as the soma. In the central nervous systems, you have this axon and dendrite configuration that we'll show a picture of in a bit, where they extend out sometimes into very long paths. The central nervous system refers to the brain and the spinal cord. The peripheral nervous system is the part of the nervous system that extends out from the central nervous system into the limbs or the extremities. A sensory neuron is a little bit different, often referred to as afferent or ascending. Basically, these pick up stimuli from the environment. The interneurons link sensory and motor neurons in the central nervous system, and a motor neuron, referred to as an efferent neuron, carries a signal from the central nervous system to the muscles. So the body has a feedback situation where you sense various things. It can be your position, it can be the ground beneath your feet, it can be something is hot and you need to move your hand away. You sense these various stimuli, and there is some communication then that occurs that then allow the motor neurons, which actually control the movement, to respond to the sensory stimuli. So here's a diagram of a neuron. The dendrite are these things that extend out from the main body of the cell, and then here's an axon. It's headed to communicate with something else. So an action potential, like we discussed in the past, would travel down this axon in a hopefully controlled way and then reach the terminals of the axon where the action potential is then transferred to make something happen. So there are three main types of nerve cells. There's sensory cell, which is illustrated here, where we're actually picking up external signals. There's a relay cell, which communicates back and forth sensory to motor. And then there's the motor cell, which is indicated by this one on the far right. So here on the far right, here's the cell. Here is the myelin sheath. Here are the axon terminals where it's actually communicating with something. The reflex arc is what's known is kind of what we described earlier. You have some sort of, let's use the example of a hot plate. So your finger touches something hot. The skin receptors perceive that, and that signal travels up a sensory neuron to a relay neuron in the spinal cord, which then connects that to the motor neuron. That's going to enable you to then pull your hand away from the hot source. So this is sometimes known as the reflex arc. This can occur without you even being cognitively or consciously aware. So your reflex to pull your hand away from a source of heat, interestingly enough, does not involve the brain. We can see if this video will play.
Reflex action and reflex arc. Reflex arc. Reflex arc consists of a receptor, a sensory nerve, an association neuron, motor nerve, and an effector organ. The receptor may be a single cell or a group of cells. They receive information about stimuli and generate electrical potentials. The sensory nerve is associated with the receptor, which carries the information in the form of electrical impulses from the receptor organ to the spinal cord. In the spinal cord, the sensory nerve makes connections with other neurons in the gray matter. These neurons are called interneurons or association neurons. The neurons process the information and generate responses. The efferent fibers of motor nerves carry these responses from the inner neurons to the effector organ, which is usually a muscle. This makes the muscle to contract.
Okay, so that's just a little YouTube video that basically says what we just said. And here is an example. Here's an example of the
Reflex action.
Here is an example of the reflex action in the leg. So I don't know if you go to the doctor. They don't do this too much anymore. But you can tap on a place just below your kneecap. It causes a strong reflex. That's their way of testing that you have healthy reflexes and healthy motor neurons. So how does this happen? This is accomplished through something that is called a motor unit. So a motor unit consists of a single motor neuron, and that neuron can activate or innervate more than one muscle fiber. Innervate means you supply an organ or a body part with nerves. So here's the neuron. When it gets ready to send an activation signal, that signal travels down the axon that's protected by this myelin sheath. When it reaches the end of its journey, it then is connected to muscle fibers. And there are these end plates that then attach themselves to muscle fibers. And you'll notice here there is some branching that occurs. So a single motor unit can send a signal to various muscle fibers. a single motor neuron can send a signal. So this entire thing, the neuron and the muscle fibers that it innervates, is referred to as a motor unit. So the innervation ratio is the number of muscle fibers that are stimulated by one motor neuron. Muscles that control fine movements, like your fingers, your eyes, will have small motor units, but then larger weight-bearing muscles, like your hips and thighs, will have larger motor units. So here's just a little different cartoon illustrating here is a whole muscle. Here is a bundle of single muscle fibers. Here's the motor neuron that's innervating certain fibers. The connection between the motor neuron and the muscle fibers is referred to as the neuromuscular junction. There are three types of muscle units that can be innervated. Slow twitch motor units have a slow contraction. There's a small amount of force. So this would be comparable to, say, a marathon runner. This is when your body sort of goes into automatic mode. Fast twitch motor unit is a fast contraction producing a lot of strong force. And type 2A might be like a sprinter. And type 2B would be the fastest twitch motor unit. This would be the strongest force. Example is boxing. Another example might be tennis. In tennis, when you serve, the better your type 2B fast twitch motor units, the stronger, better racket head speed you can get, and the stronger you can serve. Another example, as they give here, might be a boxer. So there are different ways physiologically to stimulate the innervation of different types of motor units. Some of it is dependent on your basic biology, but some of it can be modified. So in controlling a muscle force, this has to do with how many muscle fibers are recruited to do an action. So there's something called the size principle, and that occurs when smaller motor units are recruited first, and then as larger forces are required, your larger motor units will be recruited. This is the body's way of sort of controlling its own muscle system. So three types of muscle fibers, just to review, we have slow twitch or type 1. That would be a slow contraction and a high resistance to fatigue. We have a fast twitch A, which is type 2A. That would be a moderate resistance to fatigue and a transition between the other two types. And then a fast twitch B would be very sensitive to fatigue, used for a very short anaerobic and high force production. So a muscle twitch is the response of a muscle to a single action potential and its motor unit. And the fibers will contract quickly and then relax. So here's an example of muscle tension. You have a little stimulation that comes from the motor neuron. There's a slight delay, and then you have a contraction phase and a relaxation phase. So this would be just a single action potential from a single motor neuron. And so this would be when you actually have a movement, it's a combination of many of these. Here's a little bit of detail on the neuromuscular junction. This is the connection between the nervous system and the muscular system. So this is kind of a blow up of what we saw earlier. It's a kind of a cartoon. So here's a capillary or blood vessel that's supplying oxygen to the muscle. Here's the axon of the motor junction, and then here's the neuromuscular junction. And if we were to blow that up, you would see that the way that looks is that the muscle fiber has a thing that's sort of this neuromuscular, this axon fits perfectly. It's almost like a little indentation, like it was meant to be, it was meant to fit. And then this is a cell junction. And basically, the cell potential and the action potentials change the permeability, allowing for things to flow back and forth between the neuron and the muscle. So there are some technical terms here that I don't particularly care that you get those. The goal of this lecture is just to show you an overview of where the actual biopotential comes from that we would measure typically on the outside of the body. This junction here is referred to as a motor end plate. It's pretty complex. You basically This is where um, the axon of a motor neuron establishes contact with a muscle fiber. So this is where the contact occurs. This is where a whole lot of biochemistry occurs back and forth between the muscle and the neuron. Um, at that junction, there are neurotransmitters that basically communicate between the neuron and another cell, in this case, a muscle cell. This happens everywhere, but we're talking muscles. So one of the neurotransmitters is acetylcholine, or ACH. Basically what this does is this changes the membrane's permeability to sodium and potassium ion. So this is a little technical with the biochemistry, but I included this slide because we did talk about membrane permeability in the previous lecture and the importance of sodium and potassium. So what happens is the presence of ACH basically it's one of those receptors in the cell membrane that can actually change the permeability of the membrane. So ACH is released from the nerve cell and it then attaches to receptors on the muscle cell membrane. And because of this, you can then change um, the permeability of the cell. We'll go watch the little video as long as it's not too terribly long. Yeah, it's short.
Voluntary muscle fibers is a complex process which starts with the arrival of nerve impulses at the terminals of the nerves at a special site known as the neuromuscular junction.
In this video, we are going to look at what
The
happens.
impulses cause the release from these nerve terminals of tiny packets of acetylcholine, a neurotransmitter chemical. Acetylcholine then combines with receptors in the muscle membrane and generates an electrical wave in the muscle fiber itself.
I think we'll play that one more time.
Contraction of voluntary muscle fibers is a complex process which starts with the arrival of nerve impulses at the terminals of the nerves at a special site known as the neuromuscular junction. The impulses cause the release from these nerve terminals of tiny packets of acetylcholine, a neurotransmitter chemical. Acetylcholine then combines with receptors in the muscle membrane and generates an electrical wave in the muscle fiber itself.
Okay, so you could see that that is representative of some of the things we discussed in the previous lecture. So electrically what happens is the action potential arrives at this terminal, at the axon terminal, and then you have some depolarization which opens the calcium channels and calcium enters the axon terminal. This then stimulates the two membranes to fuse together, and then you have ACH, which is released and has an action on the muscle cell. So once again, you're not going to have a quiz over the details of the biochemistry, but it is interesting to understand where the action potentials originate from. Next, the ACH binds to the receptor side. This creates a depolarization, and then that allows the action potential to continue into the muscle fiber. It stimulates the release of calcium ions into the muscle. This is why calcium in the body is so important for healthy musculoskeletal system, not just the bones. We think of calcium with bones, but it's actually critical to muscle contraction. And it's sometimes when athletes will get muscle cramps, in the short term, it's often a lack of sodium, but if this is a persistent problem, they want to look at your calcium and make sure that it's present. So once again, just looking at this neuromuscular junction, here's your muscle fiber, here's your motor neuron axon, here are your motor end plates, here's a blow up of how that motor end plate joins to the muscle. So here's a little cartoon from another source that illustrates sort of an interaction. In this figure A, this is an example of where you have two motor neurons. Each one is branched. So motor neuron number 1 is stimulating these 3 muscle fibers, while motor neuron number 2 is stimulating these 2 muscle fibers. And you can see here are the action potentials. Now, for whatever reason, let's suppose that motor neuron number 2 is damaged or destroyed. What happens then in nature is that motor neuron number 1 forms additional branches called, and sometimes referred to as innervation, but it forms additional branches. And so muscle fibers 2 and 4 now have a new connection originating from this motor neuron. Now, remember that the motor neuron sends a single signal, so the disadvantage here, so the advantage here is that the muscle fiber will not die. It will get a source of excitation. The disadvantage is that it's now being controlled by the same motor neuron as fibers 1, 3, and 5. That may or may not be a problem, but you do lack a little bit of flexibility in that now you're getting a single motor neuron signal to all the fibers. So this is why sometimes after nerve injury or after nerve death, things are never quite the same. You may never quite have the range of motion that you had in the past. So let's talk a little bit then about how all this stuff is measured, because this is a biomedical instrumentation class. So you've seen that, just in summary, the biopotential associated with muscle activity originates from these long axons in motor nerves, motor neurons, and then those neurons then connect with muscle. They transmit an electrical signal. That electrical signal makes the muscle twitch or move or contract. So electromyography, I want to be sure that I cite the author of some of these slides. This was referenced earlier in the presentation, but this comes out of the kinesiology department at McGill, talking a little bit. They have some very pretty pictures. She has some very pretty pictures here, so I chose to use some of her slides for this illustration. So what is electromyography, or EMG? It's basically the technique that's used to record this electrical activity that occurs when a motor unit fires. Now, this is really important. We're not actually measuring muscle force per se. We're measuring the electrical signal that goes from the motor nerves to the muscle fiber to create force. So the EMG is basically the trace of that electrical signal that gets detected by electrodes. And the electrodes are one of two types. They're either surface electrodes, which are basically applied to the skin, or they're little needle electrodes that are implanted into the muscle. The needle electrodes are advantageous because they can pick up very localized signals, but they're invasive, and once you invade the body, then you interfere with the body. So skin electrodes, while they have some disadvantages, have the advantage that they're non-invasive. So just a little bit of history. Early on in the 1600s, people concluded that the brain basically sends a signal that causes muscles to contract. And then in the late 1700s, it was shown that electrical stimulation of muscle tissues produces contraction and force. Then about 60 years later, it was discovered that contraction and force is associated with electrical signals in the muscle. And then finally, a few years later, Duchenne was the first that actually performed systematic investigations where he used an electrical stimulation approach. Then finally, in 1985, Besmajean and DeLuca summarized existing knowledge on muscle function as revealed by EMG studies. So you can see that was 30 years ago. The technology was pretty mature by 1985. What has changed since 1985 has been the ability of microprocessors and microcomputers to actually, in real time, acquire EMG signatures and do something with it So hence the development of things like smart prosthetics and smart wearable sensors. So what can electromyography or EMG tell us? It can tell us whether muscle is active or inactive in a given task. It can tell us something about the time relationship. When does it turn on and off? Is there a time relationship between two muscles of interest? Is the magnitude of the EMG activity greater, implying higher stress? We can begin to note when muscle is fatigued, and we can sometimes also tell if the muscle is injured or diseased. And for these latter two, there are some signal processing techniques, and a current area of research is how do you identify features associated with the EMG that would give you information on muscle health and disease states. So once again, the functional unit of the neuromuscular system is the motor unit. It's comprised of the cell body and dendrites of one motor neuron, and then multiple branches, and the muscle fibers that are innervated by this neuron. Motor unit size can vary a lot. You can have as few as 5 or 10 muscle fibers that are innervated by one motor neuron, and this would be very fine motor control like eye movements. Or you can have thousands of muscle fibers in the biggest motor units. So these would be like, things that are actually providing strength to quadriceps, hamstrings, etc. The origin of the EMG signal, as we discussed before. So we've talked about a single motor unit, but now when you put an electrode on skin or into a muscle, you actually are measuring many individual motor unit action potentials because the probe or the sensor or the electrode is acting in many ways like an antenna. And so, as you know, if you have mixed signals and you have an antenna present, it's going to pick up the mixed signals. So each motor unit action potential has its own unique firing profile, and so there is some work on taking a raw EMG signal and doing a signal decomposition so that you can literally pick up individual motor units. Obviously, if you have a single sensor, doing a decomposition like this becomes very, very difficult. So what you actually have is you have usually multiple sensors, and this decomposition is done using some kind of component analysis or blind source separation technique. Here's an illustration that shows a number of motor units that fire. If you take all of those motor units and you weight them and superimpose them, then you get a signal that looks like this, which is what you actually measure by the surface electrode. So this can be difficult to analyze if you have a single electrode pair. You don't know the physical location of the motor units and which one corresponds to which firing. You don't know the proximity of where they are to the probe, nor do you know the amplitude of the individual motor units. So this can be a difficult to interpret process. You can tell things from a raw EEG. You can tell things like, is the muscle contracting or not? You can often sense fatigue from a raw EEG. Beyond that, to really ascertain anything else, you have to have a great deal of signal processing that occurs. So here's an example of what things look like. So this would be an EMG that consists of all of these weighted and summed action potentials from the motor units. You see you can get an active phase where you see this sort of contraction. You see this burst of signal, and so you see several signal bursts. Notice that there is some sort of randomized and stochastic nature to the signal. These spikes are not repeatable. So here's a person repeatedly contracting, and you can see each time the signature looks quite different. The rest period looks relatively calm. It actually doesn't look quite like this because there's usually quite a bit of noise, even at rest. So it's dynamic. The repetitive action is fairly easy to see. In the tonic or static part of the signal, activation is more difficult to see. There is always some baseline noise. The typical amplitude is in the microvolts, but it can be up to a few thousands of microvolts, especially in an athlete who has a well-developed muscle, and the frequency range tends to go from 20 to 150 hertz. So being that we're engineers, we can immediately see there are some issues here because there are several ambient noise type issues, particularly with 60 hertz, that show up right in the middle of the frequency range of interest. So what are some factors that affect the EMG? First of all, the diameter of the muscle fiber that's involved in the signal, the number of muscle fibers, the conduction velocity, the fiber type, the fiber location, the firing rate, the amount of blood flow that you get to the muscle, the distance from the electrode to the muscle fiber, the amount and type of tissue that surrounds the muscle, what's the hydration state of the muscle, are you dehydrated, the number of active motor units, the level of fatigue, and the temperature. So those are all factors that occur sort of internal to the body that affect the EMG. Go back. The extrinsic factors are factors that occur outside of the body. Characteristics of the electrode skin interface. This is what we will discuss in Chapter 5. How does the interface between the electrode and the skin surface impact the signal? Your signal conditioning. So what do you do with the signal once you acquire it? Your inter-electrode spacing. So how many electrodes do you have? How far away are your electrodes from the motor point? And then finally, the ambient noise in the room. This can be obviously 60 hertz noise or nearby machinery noise. And also crosstalk, so signals from sources other than what is studied. So your pulse, your respiration, any movement that occurs other than the actual muscle movement that you're trying to measure. So lots of kind of interesting factors associated with the EMG. Some of the most important sources of noise are tissue characteristics. So once again, we will discuss this again in Chapter 5, sort of your skin impedance, how much fat do you have, are you hairy, do you wipe away the skin oils before you make your measurement, lots of things like that. Physiological crosstalk, so crosstalk from neighboring muscles, from heart, breath, from movement of the cables if you're not wireless. Your ambient noise, so 60 hertz noise, quality of your electrodes in your amplifiers, and then just a comment that it's very important then, if you're going to acquire these signals, that you take adequate preparations in improving the conductivity of the skin, as well as in your post-processing and filtering. Here's an example of a contaminated signal contaminated with heartbeat. So here's your muscle movement. This is an ECG that's sort of showing up in the background to the muscle movement. Okay, so I just want to repeat the figure now that you've sort of seen the fact that these neurons are long. It lends a little more credence to the figure that we showed at the end of the first lecture, where you modeled these neurons electrically as a unit length. So you take some unit length of that neuron, delta Z, and then your conductance or resistance, as well as your capacitance of the membrane and your sodium and potassium sources of energy. Basically, those then are modeled in a per unit length configuration. And finally, here's the figure once again illustrating the action potential traveling along the axon. And then here it is shown with the myelin sheath. So hopefully that gives a little more insight into where the electromyographic or EMG signal comes from, as far as what's happening in the muscles. I would refer you to look at some of the sources that are referenced in Blackboard so that you have a better understanding. And if you're more interested in this, there will be an opportunity for you to look at it more. Let me just really briefly show. Here's a single motor unit potential recorded in a frog muscle. Here is an illustration of sort of how a transducer in a human muscle, a needle electrode would work. And then here's sort of what the measurement system looks like. So you're actually measuring, it's dependent on muscle length, you're looking at the velocity at which the muscle shortens, basically trying to do a control system. Look at it as sort of a control system for those of us who have some knowledge of that. Here's how the actual measurement is made, and we'll discuss this much more in Chapter 6, but you basically have a set of bipolar electrodes. What we're looking at here is we're looking at how well does this nerve actually conduct a signal. So the nerve stimulated at two different sites, and then you measure in the muscle the response. And so by looking at the delay, that tells you something about how well the muscle is actually working. Here are some action potentials after you're stimulating the index finger. So you've got a little electrode here that stimulates the index finger. You're trying to pick up that action potential as it travels up the arm. So you're doing that with these electrode pairs. And here is the same kind of thing on the foot, showing placement of electrode pairs. In this case, one is stimulated and one is picking up the stimulus. And then finally, here's just a diagram of a single motor unit consisting of the motor neuron coming from the spinal cord, stimulating the skeletal muscle fibers, and talking a little bit about the reflex arc that we discussed earlier. So your book does bring these things up. I think his figures are not quite as pretty. His explanation is not quite as clear. So I'm providing you lots of additional information if this is an area of interest of yours. But the overview that we've presented today is primarily what I think we're interested in you knowing as we proceed on to the next.